Welcome to Stem Cells Unveiled From Basics to Breakthroughs. I'm your host, Dr. Sukant Khurana. I'm a neuroscientist, cell biologist, mathematician turned into an entrepreneur and a pharmacology researcher as well. I juggle various things to find eventually solutions and hence a lot of interest with stem cells. Joining me is a very young but very promising scientist, Basudha Banerjee, who from her bachelor's days has been interested in regenerative medicine and has done various projects, research projects. She's even collaborating with us and has been working with some premier Indian institutes on various things towards stem cell biology, towards regenerative medicine. Vasudha, thrilled to have you on board.
Thank you, Dr. Khurana.
So let's start with this. Vasudha, I would say stem cells are fundamentally cells with capacity to generate diverse cell types in body, driven by two key traits: self-renewal, making identical copies of themselves, and differentiation, transforming into specialized cells like neurons or hepatocytes. It's their plasticity that makes them so intriguing.
Absolutely. So, this plasticity is what hooks me every time. I love how self renewal keeps the stem cell pool alive, almost like a reservoir, while differentiation lets them adapt to the body's needs. But let's break it down further. So, what types are we dealing with? I mean, embryonic stem cells, they come to the mind first, but there is more to the story, right? So, how do you see the spectrum, Dr. Khurana?
There are three major categories of embryonic stem cells: adult stem cells, embryonic stem cells, and induced pluripotent stem cells. Embryonic ones are from early blastocytes. They are truly pluripotent, capable of becoming nearly any cell. Adult stem cells, like those in bone marrow, aren't entirely pluripotent. They are more restricted. They are more often than not multipotent, tied to few specific lineages. Then iPSCs, induced pluripotent stem cells, those lab reprogram wonders, bring adult cells back to pluripotent stage. They have been the source of biggest like news in media. They are sort of brilliant work around, don't you think?
Absolutely. And that is totally brilliant to hear. I mean, the iPSC breakthrough that sort of blows my mind. Reprogramming a somatic cell, say a fibroblast, back to pluripotency with the help of all the Yamanaka factors, say OCT4 and SOX2, that is such a game changer. I remember when Yamanaka got published the paper in 2006, it felt like a science fiction is becoming reality. I'm sure you must have followed that development closely back then.
Yeah, I did. I was actually still a PhD student, had almost about to complete my work. There was a lot of buzz, especially in my development biology colleagues in University of Texas at Austin, as well as Northwestern. So my PhD spanned three institutes. I was doing neurophysiology at UT Austin on sort of auditory brainstem, and then doing cell biology and molecular biology in collaboration with Northwestern, and mathematics modeling neurons with NYU. I would say intellectually, probably NYU was the most rewarding part and has later resulted in me taking on things on AI. But the shift, I mean, essentially, even in UT Austin, the building we were in, Patterson, back then, I don't know where, because I know neuroscience moved to other buildings, had a lot of cell biologists, developmental biologists. For a year, this is like was a daily chatter. And I also heard back from NCBS time, some people developing interesting factors take it to like fusion from CCMB and all. So it was very exciting time. But it was built on decades of clues, right? I mean, think of 1960s, McCulloch and Till's experiments with bone marrow cells. In mice, they showed us self-renewing progenitors. Then Thompson's 1998 isolation of human embryonic stem cells. There's been a steady climb. Thompson's thing I know from my college days. 97 is when I started studying microbiology in Delhi University, and the few professors, there were two guys, Srivastav and Mehta, who were going often to US, Germany for postdocs. taking breaks and all that. So they were research inclined. So used to hear from them all these things. So it was a great time. I mean, it's the but ferment is not over yet because now whatever was built from 1960s and then Yamanaka factor to now it started to translate to real medical therapies. I mean, you can see something that's close to my heart. I hope I can help. I'm not entirely sure. Maybe there's a 5-10% chance. Is my father has lost vision in one eye almost entirely. And I am contemplating there is a French company which has also started trials in China on restoring with sort of diabetes, retinopathy related vision loss. So like where if retina is detached, in his case, if he was responsive, telling us the problem within a short time, it could have been reattached. It was done months later, so the restoration of vision has been minor. But there's stem cell thing. So now, but the thing is, back in 2008, 10, 15, these were sort of science fiction or things in lab. Now they are reaching clinical trials and in some cases being commercialized, like the vision thing, which I'm really interested in. Two, three other things I'm really interested in, and I'll not lie, they are related to family issues of even kidney and liver issues. So with stem cells, there is a lot of hope. I hope I can help my older generation, dad, his brothers, sisters, mom's siblings. My mom is gone, unfortunately. She's no longer there. But there are those, and then probably in 5, 10 years when I'll start having some of those problems, or 15 years, 20 years. So I am interested in things which closely affect me. I'm interested in things which don't affect me, but obviously most fascinated. So it's been a very steady climb, and it's very fascinating to see things reach from lab to actual therapies. It's amazing progress. Vasudha, over to you.
Yeah, I mean, that is a great perspective, and I'm sure stem cells in the near future would do some great jobs. And the timeline you mentioned about the McCulloch and Till experiments and even the Thompson's 1998 isolation. So I recall how foundational those experiments were, and one of them also showed how hematopoietic stem cells could regenerate the entire blood lineage. And then we also have Thompson's work, wherein those embryonic stem cells, they seem to be a gold mine for understanding potency. Speaking of which, let's zoom into their roles in vivo. In development, they are the architects, right? Building us from a single zygote. But postnatally, how do you see them functioning day to day?
Uh, exactly. So in embryogenesis, embryonic stem cells orchestrate everything, differentiating into germ layers that form organs and tissues. After birth, adult stem cells take the baton. Hematopoietic stem cells in bone marrow churn out blood cells, while epidermal stem cells in skin basal layer repair wounds. Their bodies sort of quite maintenance crew. I don't think I don't know enough about some of the early animals like sponges and all in this regard. But thinking of a human life form, a mammalian life form, even without few weeks of work of stem cells, is next to impossible because think of bone marrow, right? Think of our blood cells. Think of our skin, right? You cannot survive without these stem cells and stem cells with varying level of commitment, right? To pluripotency, to multipotency, to even very rigid program. But stem cells are essential for survival. They're not like some small thing in the corner. I think there's still a lot of biologists need to have this perspective.
Absolutely. And this is what makes me love stem cells. Like they're the quiet maintenance crew. In fact, in one of my research projects, I isolated mesenchymal stem cells from the bone marrow, and then I tried to differentiate into the myocytes, which would eventually help in the muscle repair and regeneration process. So anyway, it's incredible to know how they balance quiescence and activation simultaneously. And I've always been fascinated by how these niche signals, like the WNT or notch pathways, they keep them in check and even push them further to differentiate. Say for the intestine, for example, the crypt stem cells, they're constantly renewing the epithelium. So, do you think that that is a model for other tissues, or is it just too unique?
The intestinal crypts are a stellar model, rapid turnover, tight regulation. It's not universal though. Neural stem cells in brain, for instance, are far less active, more dormant unless triggered by injury. It's the diversity in behavior that keeps the field so dynamic.
That is rightly said. So, the context dependence is wild. So, tying this all together, why the obsession with stem cells now? I understand the therapeutic angle, the regenerating tissues, modeling diseases with iPSCs, but what is the big picture for you? Where do you think this foundation is taking us further?
For me, it's the promise of precision repair. Think spinal cord regeneration or lab grown organs. The foundation we have laid, understanding their types and roles, sets us up to tackle neurodegeneration, something very dear to me. Epilepsy, neurodegeneration, as you know, my first company, Iron Cure, that's what it targets. Heart failure, you name variety of diseases, right? So, what's your take on it?
See, I am with you on the repair part, but I would also add discovery to this. iPSCs let us study human development and diseases in variety of ways we couldn't even think before. So think of patient-specific models for rare disorders. It is foundational science feeding into applied breakthroughs. And honestly, I could geek about it all the day.
And we will across the series. That's a wrap up of episode one, folks. Thanks to Basudha Banerjee for bringing her expertise and enthusiasm. Next time we'll dive into more nuts and bolts of this stem cell work. You can reach out to us, type in comments, subscribe to us. We will reach back to you for every comment in the series. Thank you very much for joining us. Thank you, Vasudha, for the time and expertise.
Thank you very much, Dr. Khurana. I'm eager to unpack the further knowledge with you. Thank you.